Okay, so welcome back. It's time to have a look at determining relatedness. Would that be fast enough? So this is our third unit within Area Study 1. How do we determine how things are related? So we look at all this molecular homology now, so that we now have a lot more evidence beyond the physical stuff, the morphology and the fossils, because we can look at DNA, we look at amino acid sequences, look at the proteins present, and we now know that it's much easier to find connections. And in fact, a lot of the work done by people to create classification systems is being redone because we're now finding that they're not quite as accurate as we thought they were. And we create phylogenetic trees to show these relationships, and we'll do some phylogenetic tree creation ourselves. And then there's this interesting idea of these master genes, which are going to help solve a problem that many people have had with the theory of evolution, and we'll come to that when we get there. So, amino acid sequences. Well, before we could do DNA sequencing, we could actually sequence a protein. So get a protein and pull it apart and work out the sequence of the amino acids. And when you compare that between species, obviously if you're closely related, the amount of mutations between the amino acid sequences, well, in the DNA that's created the amino acid sequences, is reasonably low. If you're not particularly related, then there's going to be more and more differences between your amino acid sequences. And so we find ourselves quite closely related to pigs. And we can use pig proteins because the amino acid sequences are very similar. We can only use them in the short term in medical interventions. And insulin-dependent diabetics used pig insulin for a long time. That's what I need to say. That's right, enough, isn't it? So it gives us an indication of having a similarity in our common ancestors because we share that amino acid sequence. Remembering, of course, that we share the original DNA. We all have a common ancestor in the sludge, 4 billion years ago. So the mutation rate between these tells us how far apart we have evolved over that time. DNA hybridisation is another wonderful technique which has been developed. And you can actually take DNA from 2 species and we can cause that DNA to come apart. So we can separate it by heating it, and the hydrogen bonds all break. And then you put them all back together again. You put the 2 species together and cool them, allowing them to rejoin. Normally put markers in so you know which ones have got cross-species connection rather than just you put the monkey back together again. And then you then heat them again. And so the amount of heat required to pull apart those bits of DNA that have cross-species connection tells us how closely related they are. So the more nucleotides are lined up, the greater the amount of heat energy it takes to pull them apart again, and the more closely, therefore, you are related. So clearly, human and chimpanzee DNA, when mixed, shows about 98% of nucleotides are the same. So it takes almost takes almost the same amount of energy to pull apart human and chimp DNA as it takes to pull apart human or chimp DNA. So it shows that they're really closely related. So our common ancestor is not that far away. Oops. Thank you, IT staff. Go away. Okay. And of course, now we can sequence complete species DNA. And we have these genomes. We don't have all of them. There's a lot of work to be done to get all species. But a lot of species that we work with regularly are being done. A lot of disease is checked, of course, because we need to find ways to stop the disease causing illness for us. So pathogens are often sequenced. And this is now showing us an incredible amount of information about evolutionary connection because you can actually map the mutations between species. So clearly, the fewer differences in your genome, the closer you are as an ancestor. So it's sort of the next step after hybridisation. You can actually go online. There's a website in the States that allows you to do this. If we get some time, we'll go play with it. So where do we get DNA from? Well, obviously we have nuclear DNA. You have the DNA that's found within your cells. That's your own DNA. But you also carry mitochondrial DNA, and in plants they also carry chloroplast DNA. mitochondrial DNA, mitochondria and chloroplasts of being prokaryotes living in symbiosis, as we believe, with eukaryotes, are able to do their own cell replication. So mitochondria in muscles will create more mitochondria if your muscles are being worked hard. Mitochondria in adipose tissue, yeah, not so much. Don't need to work so hard. Chloroplasts will develop in parts of the plant that are exposed to light. If that part of the plant's not being exposed to light, then chloroplasts don't tend to develop at all. Or they're in very small numbers because they're not being called upon to do anything. But this DNA is really quite interesting, and particularly mitochondrial DNA is very useful in studying species, particularly within a species, and the development of phylogeny and things. And we'll do some mitochondrial DNA with marsupials and look at the phylogeny of marsupials in class. So we have this thing called a molecular clock. And a lot of people aren't sure that it's as regular as we used to believe, but essentially mitochondrial DNA is believed to mutate at a reasonably regular rate. So the amount of mutations between samples of mitochondrial DNA suggest a time back, how far back you go in time to a common ancestor. And so we can use that in a way to sort of time how far, but when the separations between species occurred. So when species arose from a common ancestor. There's recent studies that bring that into question, but it's still reasonably valid. And chloroplast DNA can be used for plants as well for the same purpose. Of course, with mitochondrial DNA, we only ever get our mothers. So because when we're conceived, the sperm essentially is a head of DNA with a tail, and at the base of the head is a bunch of mitochondria and packets of glucose for the few days it'll live. When it enters the egg, it leaves behind the tail its mitochondria. So the only mitochondria present in the new developing organism is its mother's from that egg cell, which allows us to do some interesting things with human studies particularly. Ancient DNA is obviously a source of study, especially when a lot of people are using it now, particularly with humans. We try and work out how the hell we all fit together. So it's relatively robust. It is reasonably well preserved. I mean, the further back you go in time, the less useful it is, but it can be quite useful over the sort of lifespan of human species. And so we've been using it for things like working out humans in Europe, how close to Neanderthals are we? And they're significantly different, but at the same time, we now know that Neanderthal genes are found within the Homo sapiens. So we obviously interbred or did something of that nature at some point in time. People have taken up mammals and bison and things trapped in the Ice Age. We've got thylacines from Hobart Zoo. And so there is DNA there to play around with these species and think about what we can do about recovering them and whether we should. Talk more in class about that one. Here's a phylogenetic tree. Richard Dawkins, a wonderful science writer, particularly interested in evolution, talks about these points here as being conchesters, so a common ancestor. So at that point, there is an animal, in this case, that is a common ancestor of, let's come back here, all living things, all animals, because all animals radiate out from here. At this point, we see a common ancestor between lizards and snakes. Here, a common ancestor between crocodiles and birds. I should have just there. That common ancestor there is for lizards, snakes, crocodiles, and birds. The common ancestor here, turtles, lizards, snakes, crocodiles, birds, and mammals. Okay? Us, in that blue group. The amphibians branched off here, frogs, salamanders, the lungfish are back here, rayfin fish back here. So these points are points where a common ancestor occurred. And they're really useful ways, and obviously the molecular clock allows us to try and time when these things occurred. And so we often see these things with years ago on them. And we'll do some play. We'll have some play with phylogeny. It's interesting stuff. Now, the other thing, really interesting new part of the course, in fact, this is new to the course, is this idea of the master genes. We often talk about, the Galapagos finches, Darwin's finches, but they actually answered as many questions as they answered, because they radiated really quickly, and evolution's meant to be slow. So how the hell, in a few thousand years, does a pair of, or a few pairs of finch from Chile become 13 species, filling every niche available to them on a bunch of islands? And so the discovery of these things called master genes has finally helped us unpack that one. And there are two master genes that are at work here. There's the BMP4 gene, and it determines the width of beaks, and a CAM1 gene, which actually influences beak length in the bird. It works in birds and the jaws of fishes. And so we have these two species, Galapagos Finches, and here's there's 13 species of them, and they spread over these islands, and they basically avoid competition. So these birds blew in from Chile in a storm, is the theory. It's quite a long way from Chile to the Galapagos Islands, and it would have been even longer then. The islands, being recent volcanic formations, had no resident birds, so all the niches are empty. And these two genes, these two master genes, were able to cause a huge amount of really rapid evolution in beak shape in particular in these birds to allow them to fill those niches, to become different enough to fill those niches. And over time, natural selection pressures and changes cause them to stop interbreeding. And so they are now recognised as 13 species. They are incapable of interbreeding, and they are now even looking different. Big difference is the biggest thing. It's about food, which is obviously our selection pressure at work for them. And interestingly enough, in Africa, we see something even bigger. It's even huger, if you like, than the fish. And these are than the finches. These are cichlid fish. We've got some cichlids in the back of the biology room. So this is one lake, Lake Moali, and these are all cichlids that live in Lake Moali. These are the cichlids of Lake Tanganyika, and all these islands, these lakes are here in sort of central Africa. And over time, during ice ages and interglacial periods, they've gone completely dry and then filled again. And every time they fill again, the cichlid fish appear, probably eggs or a few hang on in small pockets of water somewhere, and radiate out and fill all the possible niches. And amazingly, it's still the BMP4 and CAM1 genes acting as master genes, impacting a whole range of other genes that create the differences in fish jaw that allows them to use different niches. And look at the huge variety. This is bigger than the Galapagos finches for two reasons. One, the array of species is enormous, but two, it's happened a number of times. Every time these lakes dry out, they appear again when they fill up. So it happens regularly. This is the sort of thing that would, put the wind up most evolutionists because it happens too bloody fast. But these master genes are suggesting a way of this occurring and putting some interesting future thoughts about other species. And so they're going to be interesting to see how these, what sort of research is done with these in the future. I think that's the end. It is the end. So I'll stop there and start another one.